What the Study Says.
Welcome back to What the Study Says. I'm Dr. Kai.
And I'm Dr. May.
What if I told you that some of the most genuinely surprising scientific breakthroughs actually started with, well, a good laugh, and maybe some really wiggly creatures?
Today, we're doing a deep dive that I think perfectly captures that spirit. Curiosity, unexpected discovery. And it leads us to one of science's more delightful honors, the Ig Nobel Prize.
Right. For anyone who hasn't heard of it, the Ig Nobel Prize, it kind of celebrates the unusual and honors this imaginative research that first makes people laugh, then makes them think. It's about finding real science kind of hidden behind these often hilarious ideas.
Exactly. And our focus today, it's the, well, incredibly witty, but also surprisingly deep 2024 Ig Nobel Chemistry Prize winner. You ready for this title?
Lay it on us.
Chromatographic separation of active polymer-like worm mixtures by contour length and activity.
Wow. Okay, that's a mouthful. Wiggling worms in advanced chemistry. You weren't kidding.
It sounds totally wild, I know, but it's real science.
So when I first heard active polymer, my mind went to something microscopic, right? Something moving on its own power. How do they actually define that here? Especially using worms, of all things.
Yeah, that's a great starting point. So an active polymer is basically just interacting agents, things that convert energy from around them to mechanical force, movement.
Like bacteria or parts of cells?
Precisely. But for this study, they picked living Tubifex worms, these thin little guys, and it turns out their segmented bodies, they're actually a pretty good natural model for, synthetic polymers.
Huh. That's fascinating. Okay, so if they're like polymers, how did the researchers control them? I mean, how do you get worms to wiggle more or wiggle less on demand?
Well, they found two pretty clever ways. First one's straightforward, just changing the water temperature.
Okay, colder means slower, I guess.
Yep, lower temps, less active worms. Simple enough. But the second method, this is where the Ig Nobel humor comes in. They temporarily put the worms in a 3 to 5% ethanol solution.
Wait, ethanol, like alcohol. They gave the worms a drink.
Essentially, yes, just a little dip.
Yeah.
And it worked. It significantly, reduced their wiggling, their activity level.
Wow. Okay, that's dedication. So they had chilled worms and slightly tipsy worms. For science.
Science.
All right, so then the experiment itself, this scaled up chromatography channel, I was picturing something tiny, but scaled up implies bigger. How did that work?
Right, so think of standard chromatography, but bigger. They built this channel, and inside it was an array of hexagonal pillars. It's designed to mimic how scientists usually separate conventional polymers.
Okay, like an obstacle course.
Exactly, an obstacle course for worms. They then had water flowing through, carrying the worms along, and they timed how long it took each worm to get from start to finish. That's the elution time.
Got it. So after all that set up, the ethanol, the pillars, what did they actually find? Did the bigger worms still take longer, like regular polymers?
Yeah.
Or did the wiggling change things?
Both. And that's the really cool part, the dual discovery. So yes, just like regular polymers, size mattered. Longer worms, what they called greater contour length, basically just their body length generally took longer to get through. No big surprise there.
Okay.
But, and this was the key finding, the travel time also depended really strongly on how active the worm was. Higher activity meant they got through faster, significantly faster.
Faster. Really? That seems backwards, doesn't it? To get more wiggling might get them tangled up or something. How does wiggling more help them navigate?
Yeah, it's counterintuitive at first glance, but it comes down to the shapes they make, their conformations. The highly active worms, like the ones in warmer water, they were constantly changing shape, curling up, even forming little knots temporarily.
Right.
And these dynamic shapes actually helped them maneuver around the pillars more effectively. They could sort of, squeeze through or wiggle past obstacles.
Ah, okay. So the less active ones.
The less active ones, the cold ones, or the ethanol-treated ones, they tended to stay more stretched out or adopt fewer of those helpful twisty shapes. This made them much more likely to get snagged or wrap around a pillar, what the researchers called trapping modes. And that obviously really slowed them down.
That makes perfect sense when you put it that way. So it's not just random wiggling, it's effective wiggling leads to faster travel.
Exactly. And they didn't just theorize this, they demonstrated it beautifully.
Yeah.
They took a mix of worms. Some were highly active, and they dyed those red. Others were made less active with the ethanol treatment, and they dyed those blue.
Red versus blue worms, okay.
Then they put the whole mix into the channel at the same time. And sure enough, clear separation. The active red worms started coming out first. Then there was this noticeable gap about 40 seconds or so,
Wow.
before the less active blue worms finally started to emerge. It was a really clear visual proof of the concept, separating them purely by activity level.
That's incredible. So this isn't just a funny experiment about worms, is it? It sounds like it could have some really serious applications.
Absolutely. Think bigger picture. This principle could potentially be used for sorting all sorts of active biological agents, maybe in medicine, like assessing bacterial pathogenicity, how actively infectious they are, or even sperm motility for fertility treatments.
Huh. Haven't thought of that.
And maybe even environmental stuff, like designing systems where active microbes could help clean polluted water, separating the active cleaners from the duds. It just gives us this great versatile model system to study how active things move in complex spaces.
It really broadens the scope, from tipsy worms to potential medical breakthroughs.
It really does. This whole deep dive into, worms sorting themselves out by sheer wiggling power, it honestly makes you wonder, doesn't it? What other everyday things we might overlook, could hold the keys to complex scientific problems if we just, well, look closely enough.
That's a great thought to end on. And that's another fascinating deep dive from what this study says. It just goes to show how sometimes a bit of laughter, a bit of the absurd, can really open the door to some groundbreaking science.
Couldn't agree more.
We really hope you enjoyed this journey into the wiggly world of worm chromatography. If you did, please do subscribe and maybe share this deep dive with your other curious friends. We'll see you next time.